What's up guys, BIOBEAN here. So this week is reading week, so I'm going to try and pump out as many videos as I can, so stay tuned for those. This week we're going to be talking a lot about meiosis, so let's get started. Alright, so I wanted to start the discussion about meiosis by talking about where and when it happens. So they always say that meiosis happens in our sex cells, but where are they? So it depends on whether you're a male or a female. So if you're a male, your sex cells are contained within your testes. And if you're a female, then your sex cells are contained within your ovaries. And these are going to be the sites where meiosis occurs. So when does meiosis occur? This depends on whether you're a male or female. So if you are a male, then meiosis is going to happen when you start puberty. So that is when your sperm cells are going to start to be produced and you are able to be sexually active. Whereas for females, meiosis begins during development. So when you're a fetus developing in your mother's uterus, you are beginning to enter into meiosis. So your ovary cells are entering into meiosis 1 and they arrest at the end of meiosis 1. Then once you hit puberty, those then enter into meiosis 2. However, they then pause at metaphase of meiosis 2. And then every month, one of those eggs is going to finish meiosis and then it is going to drop and become an egg that has the potential to be fertilized. So they complete meiosis every month where only one egg is going to actually complete that meiosis each month in order to maintain a store of eggs throughout your life. So for males, meiosis is going to be happening within the testes and is going to be happening once the males hit puberty. Whereas for females, meiosis is going to occur within the ovaries and it is going to begin during development as a fetus. It is going to pause until we hit puberty. Then once we hit puberty, each egg is going to, on a monthly basis, finish meiosis in order to undergo the cycle. Alright, so we just went over when meiosis occurs. Let's go over what is meiosis. So here we have a normal sex cell. So this is going to be either in your ovary if you're a female or in your testes if you're a male. And so this cell is going to be sitting here and this is going to be before any meiosis takes place. So this cell currently is sitting at 2N and 2C. So in this cell, we only have a single chromosome. So it's 2N because we have chromosome 1 and there's 2 copies of them. And we know because there's 2 different centromeres. And it is 2C because we have 2 copies of them. So it is double the amount of DNA than would be in a haploid genome because we're looking at a diploid genome that we have in humans. So before meiosis can occur, it needs to undergo replication. So in the same way for mitosis to happen, you have to replicate the genome. In order for meiosis to happen, you also have to replicate the genome because otherwise information would be lost. So in replicating the genome, we move from 2N and 2C to 2N and 4C. So the coefficient of N remains the same because we still only have 2 centromeres for the chromosome 1, but the C value increases from 2 to 4 because we have double the amount of DNA that we have within the cell. So the whole process of meiosis is going from this 2N 4C cell into a 1N 1C cell. This is because if we had a 2N egg meet a 2N sperm, we would get a 4N cell. And that would just continue to increase and increase every time you had a baby. And so in order to remain a diploid organism, our gametes need to be a 1N so that when we actually go to make the zygote, we will create a 2N baby and that will remain as a diploid. So meiosis and mitosis have a lot of similarities. In mitosis, it goes through the stages of PMAT. So prophase, metaphase, anaphase, and telophase. So I like to remember them using that acronym PMAT. So meiosis essentially just does mitosis twice. So it goes through that prophase, metaphase, anaphase, and telophase 2 times within meiosis. So we break those 2 parts into meiosis 1 and meiosis 2. So let's start with meiosis 1. So here we have our sex cell, which is either in your ovary if you're a female or in your testes if you're a male, and it has been replicated. We know because it is at the 4C level. So if it was unreplicated, it would be 2N and 2C, but because it is replicated, it is 2N and 4C. So here we have 1 chromosome from your mother and 1 chromosome from your father. So when we get down to the gamete level, eventually each cell is going to either have a copy of chromosome 1 from your mother or a copy of chromosome 1 from your father. So it separates out into the different gametes that you can make. So we start meiosis 1 at prophase with our replicated chromosomes, and they are going to have arranged properly in the chromosome format. The nuclear envelope is going to dissolve and just get all ready for meiosis to occur. So metaphase 1 is where meiosis begins to differ from mitosis. So in mitosis, normally we would have these replicated chromosomes. They would just randomly assort within the metaphase plate or along that middle of the cell, and then they would get pulled to either end, where both copies are going to be retained in both daughter cells that occur after the end of mitosis. But it's a little bit different in meiosis. So in meiosis, during metaphase 1, we actually have the homologous chromosome pairs line up together. So here we see one pair of chromosome 1 is on one side of the metaphase plate, and the other copy of chromosome 1 is on the other side of the metaphase plate. So what that tells us is when this cell goes to split apart, one cell is going to get the whole copied chromosome 1 from their mother, and the other cell is going to get the whole copied chromosome 1 from their father. So to finish off meiosis, it moves from metaphase into anaphase, where the chromosomes are going to be pulled to either end. Remember, these are the replicated chromosomes. The whole replicated chromosome is going to get pulled to either end of the cell. And then telophase is going to happen, where you are going to get a pinching off and a creation of 2 cells, where one creates a copied chromosome of your mother's cell, and one holds the copied chromosome of your father. So now we're going to follow these 2 cells into meiosis 2. All right, so now we're at meiosis 2. So the 2 cells that were created at the end of telophase in meiosis 1 are now sitting at 1N and 2C. So how did we get from 2N and 4C to 1N and 2C? So originally it was 2N and 4C, and that was because we had 2 centromeres that belonged to the same type of chromosome. They were both for chromosome 1, so that's where that 2N came from. So now, because they are split off into separate cells, it is dropped down from 2N to 1N, because now for chromosome 1, there's only a single centromere that we can count in each cell. And now, instead of having 2 copies of those homologous chromosomes, you only have 1 copy. But because they contain 2 sister chromatids, that C value is going to be 2. So we're at 1N, 2C, and we have to get down to 1N, 1C. So that's going to happen by the end of meiosis 2. So now we're going to enter into metaphase 2. So now we only have 1 set of each chromosome, however they're duplicated. So now each sister chromatid is going to be pulled to either end of the cell. So to accomplish this, each chromosome that is copied is going to line up along the metaphase plate. So whereas in metaphase 1, you had a homologous chromosome on either side of the metaphase plate, now all of the chromosomes are just going to line up along that metaphase plate in a single file, because now you're just going to be pulling each sister chromatid to either side. So once the chromosomes line up single file along that metaphase plate, anaphase then occurs, where each sister chromatid is going to be pulled to either end of the cell, and then telophase is going to occur, which is going to split off each of those cells, creating 4 cells, each containing only a single copy of that chromosome. So it is going to be 1N and 1C for all 4 of these cells, which are now mature gametes that can go on to fertilize or to be fertilized. At the end of meiosis, we get 4 cells that either contain a chromosome from your mother or from your father. So this increases genetic diversity a little bit, because one of your chromosomes from your mother is then going to meet your partner's chromosomes, maybe from their mother or father, and so you're going to get a bit of mixing of genetic material, which is kind of the whole point of having sex, is to increase our genetic diversity. But we actually have a process within meiosis that allows for an increase in genetic diversity, and this is called recombination. So this is something I've already touched on a little bit when I was talking about copy number variation in the mutations video, but we're going to get a little bit deeper into it today. So while the chromosome from your mother and the chromosome from your father contain the same type of information, the exact information contained within it is going to be slightly different. So here we're going to pretend looking at a chromosome. Let's call this chromosome 1 because that's the chromosome we've kind of been running with. And they both contain a gene in the same position because they are both the same chromosome. And so both are going to be genes that code for eye color. Again, I'm making this up. I don't know that eye color is contained on chromosome 1, but we're going to pretend like it is. So both genes are going to code for whatever eye color the baby is supposed to get, right? Because these two are going to meet together and create a zygote, which then forms a baby. So both contain information about eye color, but the mom's gene for eye color is going to code for blue, while the dad's gene for eye color is going to code for brown. So while they both contain the gene for eye color, they differ in their alleles. So alleles is the type of eye color that you would create. So the type of gene that is there. So the allele contained within the mother is green eyes, and the allele contained within the father is brown eyes. So the allele is the very specific exact information that is contained within the gene. So the gene is going to tell you what the kind of general category is. So this gene is in the general category of eye color. So it's going to dictate what eye color the baby gets. But the particular allele is going to dictate which color it's going to get. So if you get the mom gene, it is going to get green eyes. And if you get the dad gene, it is going to get brown eyes. So it gets a little more complicated because there is allele dominance, which if you took grade 12 biology might be a little bit familiar to you. Incomplete dominance, complete dominance, things like that I believe you will touch on in later videos. So in the end of meiosis, we create a single cell that either contains the allele for green eyes or is going to contain the allele for brown eyes, depending on whether you get your chromosome from your mother or your chromosome from your father. And then that is going to meet a sperm that also either got a chromosome from its mother or father, and that's going to contain the different allelic information. So maybe the sperm is going to contain the allele for brown eyes, and maybe the egg is going to contain the allele for brown eyes also. It's going to create a brown-eyed baby. So there's a way for us to actually increase the genetic diversity that we see, and that is through a process called recombination. And so what recombination does is it takes the same chromosomes, so a chromosome 1 from your mom and the chromosome 1 from your dad, and it is actually going to swap the alleles contained within it. So it's going to retain the gene information because it's the same gene from mom and dad. They both code for eye color, but the alleles are going to get swapped around. So instead of sitting on the mom chromosome, the green eyes, we're going to swap it so that on the mom chromosome, it sits brown eyes. So the brown eye allele is going to be what's on the mom chromosome. So I've already touched on recombination a little bit in my previous video about mutations where I talked about unequal crossing over, but we're going to get a little more in depth about how recombination happens. So recombination takes place during metaphase 1 of meiosis 1. So this is when there are two pairs of homologous chromosomes, and during metaphase, they're all nicely lined up along that metaphase plate. And so this is an optimal time for recombination to occur because all the chromosomes are super tightly wound and they're well organized in their chromosome pairs. So in this case, we have our chromosome 1s because that's the one we always use, and they are properly lined up along this metaphase plate. So now recombination is going to occur. So I've kind of labeled the genes that are contained on each chromosome as A and B, and I've kind of marked them with an N for the mother and a B for the, or rather a D for the father. So now recombination is going to take place. So what that means is literally the red chromosome is going to swap its backbone with the blue chromosome. So they're literally going to cross over. And it is going to end up with a chunk of the blue chromosome on the red chromosome and a chunk of the red chromosome on the blue chromosome. So now crossing over is complete, and now chromosome 1 contains the alleles from the dad's chromosome. So now they've swapped the alleles that are contained within it. And this is a way to increase genetic diversity. So now when these go to make gametes, This part of the chromosome is going to contain information from both the mother and father. And so you get a mixing of alleles, and this increases genetic diversity and decreases the susceptibility to disease, or rather the chance for susceptibility to disease, as well as different genetic defects that may occur because of the defects that may be seen within individuals' genes. So we already talked in a previous video about how this recombination can go wrong and lead to copy number variation. So if these don't properly line up, that may lead to more information being transferred to one chromosome and less information being transferred to another. If you'd like to check that out, please check out my previous video, Summary of Mutations. So this concludes our first video on meiosis. Stay tuned for future videos about non-disjunction and alternation of generations in the different kingdoms of the world. And please don't forget to comment, like, and subscribe in order to continue receiving updates about these videos as they get posted so you can better understand biology.